This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 259, recorded on February 3rd, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schechter.
Well, hello there. How are you doing?
I'm very well. And yourself?
Doing fine, thank you.
Is the weather beautiful out there? Is it snowing?
Of course, it's gorgeous. Clear blue sky. It's been this way.
Here in the Northeast, we have snow, and also throughout the U.S. Also joining us from Northwestern Michigan, Michelle Swanson.
Yes, where we also have snow, but I can report the days are getting longer.
Yes, incrementally longer. And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. We have Chamber of Commerce weather. It's 73 degrees, sunny, and bright skies, and the days are indeed longer. It's now light past 6 o'clock. So we're turning the corner, and we're moving into springtime. But on Saturday, we had snow flurries, which there is no bread to be found anywhere in the town. They've consumed it all.
That's just funny. No bread
and the milk
eggs.
Is gone, too.
Milk, too.
Yes, milk too. The eggs, people have chickens, so we don't have to worry about eggs too much.
Our guest today from the University of Puget Sound, which is in Tacoma, Washington, Mark Martin. Welcome back.
Oh, it's such a pleasure to be here. The weather here is cool and rainy and green. And I'm pleased to tell you that our case numbers are starting to ramp down. So I'm teaching two classes in 2 laboratory sections while masked. We're all hopeful.
Well, this is, I've lost count of your number of times on TWiM. So thank you for coming back. We always like to have you with your high energy level. All right. Today we have, for your listening and learning pleasure, 2 papers, and Michael is going to start us off.
So this is a paper that's a little bit different, but I think you'll find it enjoyable. The title is The Global Burden of Bacterial Antimicrobial Resistance in 2019, a systematic analysis. It was electronically published in The Lancet on January 20th, 2022. The authors are the Antimicrobial Resistance Collaborators, where the first author is Christopher J.L. Murray, and the last of the approximately 125 authors was Moshe Nagavi. So if I read their names and affiliations, I would exceed my time, and we would not have an opportunity to reflect on this fine paper.
Actually, Michael, I got in touch with the authors, and if you could just give me about 40 minutes, I just want to go through the bios.
Okay.
I tell you, it was 3 pages of the paper just in like 6-point font listing all of that. But it's really, when you get a sense of what they did, you understand why they needed 125 people to do this. So let me start by saying that their conclusions were not surprising to any of us in microbiology. And I believe that many of our listeners are well aware that antimicrobial resistance is a significant issue and a major threat to human health in both the developed and developing countries. And in point of fact, it's so unfortunate the toll taken by antimicrobial resistance on patients, their families, and for the most part, this is largely invisible, but it's reflected in prolonged bacterial infections that extend hospital stays and cause needless deaths. Further, as you might suspect, antimicrobial resistance disproportionately affects poorer individuals and poorer countries who have little to no access to the so-called second line of antimicrobials, which are often newer or more selective in their spectrum of activity as a consequence of either expense or availability, And these second spectrum drugs could greatly reduce or knock down the infection as the first line drugs failed due to the resistance of the microbe, thereby failing to arrest the infection. First, this paper presents the most comprehensive estimate of AMR or antimicrobial resistance burden to date. All I can say is wow. And this snippet is not going to do it justice, but I would suggest that you do a deep dive to gain the perspective that these 125 authors have labored to provide. The maps, which are figure 7, which goes on for 4 or 5 pages, gives you a sense of the risk you face if you really want to be a globetrotter. So what we have here is they estimated the number of deaths and disability adjusted life years, or dailies, also known as disease burden attributable to AMR, to us non-epidemiologists, and associated with bacterial AMR for 23 pathogens and 88 pathogen drug combinations from 204 countries and territories from before the world went to hell in a handbasket of 2019. Now, to give you a sense of the scope, they collected a lot of data from systematic literature reviews, hospital systems, surveillance systems, and other sources that covered, now hold on, 471 million individual records or isolates that totaled up to 7,585 study location years. So this is a lot of data, folks. They then used a predictive statistical modeling suite to generate estimates of AMR burden for all the locations, including those areas where they had no data, where they needed to do a process called data imputation. Now, to give you a sense of the scope of the data that we're going to see, let's jump to one of their data slides before I tell you how they did things, by jumping to figure 3. Here they estimated the disease burdens associated with and attributable to. So they're looking at 2 different questions for the 12 major infectious syndromes. And the graph is organized really nicely for those not familiar with a lot of fancy epidemiology. The highest number of global deaths were for lower respiratory infections and all infections in the thorax. And now that makes sense. Any, casual survey of microbiology or infections, we always tell our students how bad pneumonia is as an infection. So this is where pneumococcal pneumonia, other drug-resistant pneumonias are classified. And then as they go down the list, as the number of infections descend, they go through bloodstream infections is number 2, no big surprise. Then they go to peritoneal and intra-abdominal infections. Anyone who's ever watched a John Wayne movie knows that if you're gut shot, you'll likely die. And so this, again, tells you the significance of those types of infections. Meningitis, another one, casual observers of micro know is not a good infection to get. And other bacterial central nervous infections. Then they go down to the enterics, talking about all the diarrheal types. But the one thing that really struck me is urinary tract infections. And that really troubled me because most folks don't think of UTIs as major. You go see your doctor, you get a script, and it's gone in 2 weeks. But here, if you look at the number of deaths associated with resistance to urinary tract infections that were associated with resistance, you get about 250,000 global deaths from a UTI. To me, that's way too many folks dying from urinary tract infections, where they literally attributed it to the resistance, the better quality data. It was just a little less than 100,000. To me, as a microbiologist, that's still too many individuals dying of urinary tract infections. So now, yes.
Does
Michelle.
that reflect in part that there's such a large burden? It's such a common infection. The denominator is very large, so the deaths is also large.
Yes. And we're going to talk a little bit about that when we get to another factoid I want to bring up, namely the global resistance in tuberculosis. And I'll get to that in a second. But first, I want to tell you how they developed the data that give us these really cool figures. And it'll also help you appreciate the significance of the labor they had to do in order to get these data. They parsed their study, or took the data, into 5 broad components. The first was the number of deaths where infection played a role. The second component was where the proportion of infectious deaths attributable to a given infectious syndrome was present. The third was the proportion of infectious syndrome deaths attributed to a given pathogen. The fourth was the percentage of a given pathogen resistant to an antibiotic of interest. And finally, the excess risk of death or duration of an infection associated with resistance. So they had a wide variety of data because it speaks to the larger question that we have no global database that actually records all these data. So they had to go out and hunt them up. But then using these 5, if you will, buckets of data, they then estimated the disease burden based on 2 counterfactual scenarios. Recall that a counterfactual scenario is when a factual prior event is modified and then assessed again for the consequence of that change or delta. And the 2 counterfactuals that they used, one was in which all drug-resistant infections were replaced by drug-susceptible infections, and one in which all drug-resistant infections were replaced by no infection. So without getting into the statistical weeds, and they go into a great treatise on how they did things, there's supplemental information, and the Lancet, recall, before you can publish a statistical paper, they have their own independent statistical evaluators to validate your method. So this is a really rigorous process to get the data into print. So putting their estimates into context, they found that antimicrobial resistance is a leading cause of death around the world, with the highest burdens, and again, no surprise to us in microbiology, the highest burdens in lower resource settings. Putting their estimates into context again, they estimated a median number of deaths at 1.27 million, with a 95% uncertainty level of between 0.9 million to 1.7 million in 2019.
And that's in 1 year, correct?
That's in 1 year, 2019.
1 year.
And this was directly attributable to resistance. And this is the thing that slapped me in the face. A value that is nearly the same as the global HIV deaths, which were 680,000 in 2019, and the malaria deaths, which were 627,000. Both of those are big killers. And we're killing more people because of AMR than those 2 combined. And unfortunately, it ranks behind what Vincent has been spending his time on these last 2 years, only behind COVID and tuberculosis. Now, recall that tuberculosis remains a global pandemic. This disease is killing someone approximately every 22 seconds. And in 2019, that was about 1.4 million people in terms of global deaths from a TB infection. Now, when you jump to one of their figures, looking at tuberculosis and the global AMR deaths, it's about a quarter. A quarter of the tuberculosis deaths are a consequence of antimicrobial resistance TB, for which we have no drugs. And that is really a major concern because, remember, TB has been with us for thousands of years. It's a very debilitating disease. It's a disease that spreads very easily. The infectious dose is about 10 microbes. So the fact that a quarter or more of the tuberculosis deaths are due to antimicrobial resistant TB should really give us pause and tell us we need to get busy on either a tuberculosis vaccine or finding better drugs for TB or a better treatment scheme for TB.
They also do another layer of analysis, which is they show us a map of the world and show us what the resistance rates for MTB infections are in different regions.
And that's another reason I, in the beginning, said, plan your vacations accordingly. Again, understanding the risk. Remember, your risk for acquiring tuberculosis is breathing, very similar to acquiring COVID. Your risk. The risk of acquiring HIV is, of course, it's an STD, so or bloodborne products. So again, the study offered that it was estimated that there's about 5 million deaths associated with bacterial AMR globally in 2019. And this indicates that there are substantial gains to be made from preventing infections in the first place. As this is TWiM, rather than a course in epidemiology, we're going to talk a little bit about the 6 leading pathogens for deaths associated with resistance. And I was shocked that it was E. coli at number 1. I would not have anticipated that E. coli would be up there. It was then followed by Staph aureus. Klebsiella pneumonia, I thought would have been higher. Strep pneumo, we know that penicillin works really well against most forms of strep pneumo. I would have thought Acinetobacter baumannii or Pseudomonas would have been higher, but E. coli was killing about 1 million people attributable to antimicrobial resistance. And again, as I said, this is all laid out beautifully in Figure 4. I mean, it just goes down, and you can find your favorite organism and see where it is in deaths attributable to antimicrobial resistance.
And again, that doesn't mean E. coli is the most threatening. It's just the largest burden
of antimicrobial resistance. And this makes some sense intuitively because E. coli does a lot of horizontal gene exchange. It lives in a very rich microbial world in our digestive tract where it's exposed to many different opportunities to acquire genetic information. So it's not that surprising, as opposed to Staph aureus, which lives on our skin and our upper respiratory tract. So it again, it's something that a microbiologist would intuitively say, yeah, I can believe that. But in terms of someone who follows infectious diseases and you hear all the train wreck stories, many people don't talk about the train wrecks that are E. coli. So another figure you may wish to spend some time with is Figure 6. Here they dig into the pathogen drug combinations and the global deaths attributed to AMR. Now, this one didn't surprise me. One drug pathogen combination that stuck out like a sore thumb was the methicillin-resistant Staph aureus, caused more than 100,000 deaths just due to this one drug-bug combination. And that's a really fun figure to look at. You look at the standard drugs that many of the developed world's formularies have in, and they give you the abbreviations, everything from 3rd-generation cephalosporins and 4th-generation cephalosporins, the Waxman class of antibiotics, the aminoglycosides like streptomycin, gentamicin. They lay it all out. But sticking down there, little old methicillin, 121,000 deaths due to Staph aureus. I mean, it's just
1 year.
In 1 year, folks, in 1 year. And of course, the drugs that make the CDC's hit list are, carbapenem resistant, Acinetobacter baumannii, fluoroquinolone resistant E. coli, carbapenem resistant Klebsiella pneumonia, the 3rd-generation cephalosporins. It's really an interesting figure to look at. So why did they do this? Why was there need for this study if I'm telling you it's not surprising? Well, in 2017, the WHO published a priority list for developing new and effective antibiotic treatments. If you will, it was a call to arms that was intended to inform research and development priorities related to new antimicrobials and put the emphasis on the pathogens with multi-drug resistance that cause severe and often deadly infections in healthcare. And something that we all need to be concerned about is long-term care facilities, because the globe is aging and the globe is very mobile, and many people, when they're no longer able to care for themselves, they go into these long-term care settings. And the authors offered that the intention of the WHO list was to set new antibiotic research and development priorities rather than identify the most burdened pathogen. Again, investigator-led discovery, rather than finding a utility and dictating priorities, because it's hard to dictate priorities because this is really the first comprehensive assessment of global AMR burden. So this paper is critical to helping pharma and governments answer the age-old question, will the juice be worth the squeeze? And helping funding agencies address how they should prioritize their RFAs that they're putting out. And as many of the listeners in the United States will know, on February 2nd, President Biden relaunched the Cancer Moonshot, aiming to reduce the death rate by cancer by 50% in 25 years. I'll put in my plea that the data in this one paper argues strongly that we don't have 25 years to wait to really address antimicrobial resistance, as the microbes and antimicrobial resistance seem to be operating on an accelerated timeline, their own timeline. We need a moonshot right now to understand more about the molecular pathogenesis, how bacteria interact in these complex microbial communities, and whether or not, we don't need the nuclear weapons that are antibiotics, but we really need to learn how to tinker with the microbiome, and maybe that will lead to better strategies for health, and then reserve the antimicrobials for when we desperately have that life-threatening infection. And consider that of all the major bacterial pathogens covered in the study, only one, pneumococcal pneumonia, is preventable through vaccination. So do we need a moonshot for a TB vaccine, considering what we have seen with the XDR and MDR TBs? And what about developing vaccines for viral pathogens? We have, a vaccine for influenza, but it's not all that effective, and often influenza breeds secondary bacterial infections. Pfizer is said to be committed to developing a respiratory syncytial virus for which bacterial infections are a secondary consequence. And then, of course, there's rotavirus, because diarrheal diseases are big, and there are two effective vaccines in the U.S. for rotavirus, the Rotatec and Rotarix, and they go after rotavirus 5 and 1 respectively. So could they be effective in reducing the need for putting antibiotics into the mix? Anytime you can take an antibiotic out of a treatment scheme, you're reducing the selection pressure. And the students of microbiology remember that antibiotics are tremendous selection pressure because it's either live or die. And that what doesn't kill you makes you stronger. And that's what the microbes have been operating on.
Michael, what's the reason why there are so few bacterial vaccines and many antibiotics?
My guess is it's liability in the United States and economics in other countries.
Well, and also many of bacterial infections are successfully treated with antibiotics, but they're also so widespread that the total burden and total number of deaths is high. So, and to put a sharper point, Michael, on your excellent point that this study can really drive policy decisions and prioritizations. They make the point that right now we're investing about $50 billion each year in HIV research, but the number of HIV deaths is about 900,000, which is lower annually than the number of deaths due to antimicrobial resistant pathogens. So we need to look at the balance sheet and decide where our research dollars should be invested.
It really, and that's the closing point I was going to make. The other data element to consider, and this is really in the realm of public policy, where microbiologists can inform their elected officials. Recall that in high-income countries, improved water and sanitation, public health, and hospital infection control methodologies have been the primary ways in which infections have been controlled and reduced. I mean, just imagine Florence Nightingale and Ignaz Semmelweis and their contributions to improving healthcare. But these methods have been a heavy lift to implement in resource-poor settings despite economic progress. And when I mean resource-poor settings, I mean in some of the cities in the United States, in addition to some of the developing countries around the world. I mean, you just need to look at our aging infrastructure with terms of water and sewage treatment plants. So really, this paper goes a long way in helping us understand the burden of AMR and the leading drug combinations contributing to AMR. And these data are going to be crucial to making informed and location-specific policy decisions, particularly about infection prevention and control programs and access to essential antibiotics. And again, we're all going to have to work together because this is a global and not a local problem.
And I'd like to also just thank the Bill and Melinda Gates Foundation and Wellcome Trust, who together funded this really massive effort that really could reset policy on infectious disease treatment and research.
Can I make a quick comment?
Mm.
So this is like a very, very big deal to me. I could talk about the original microbial OG Pasteur had actually said in the final analysis, the microbes have the last word. And they do. And that's important to remember. And as I said, what I'm doing in my class, and a shout out to my Bio 111 students, if they're listening, you get an extra credit point if you're listening right now, by the way. What I would tell you is that we wanted to do a course-based, genuine learning experience for them. And we're focusing on Joe Handelsman's tiny earth crowdsourcing technique to look for more antibiotic-producing microbes from the soil. So in order to get them ready for that, because it's bothered me for many years that the single most important thing in introductory biology, antibiotic resistance, we don't talk about. And so I insisted on this for years, and finally I got permission to do it. I got trained by the Tiny Earth folks in Madison, Wisconsin. I finally got approval to teach it. And the first thing I have the students do is watch that really alarming TED Talk by Marin McKenna and read some of her essays. I actually had them read the short version of this particular essay, poem that you're talking about. And to a single person, all of these students are saying, I'd never heard this before. I didn't know this. So, I mean, part of the solution here is just educational.
And I don't think our elected officials are aware of the significance of the problem.
No, I agree.
And it's not just deaths, although we care about that, but it's also a huge strain on our healthcare system, something we've learned to appreciate during the pandemic. So healthcare dollars and strain on our resources. And
the new antibiotics are not cheap. They're in, if you look at the costs, and everybody was up in arms when a couple of the drug companies raised the cost of tetracycline. And the cost of tetracycline went through the roof. There was a time when the bottle that the pills went in was more expensive than the cost to make the drug. And today that's no longer the case because the companies have figured out they can get more money for it. So we really need to begin our efforts. So I commend them. And again, thanks to Bill and Melinda Gates Foundation for underwriting this effort.
Thank you, Michael.
Thanks.
And now we will put Mark to work.
Well, the first thing that I want to say is I'm delighted to have the opportunity to talk a little bit about marine microbiology, a little bit about some wonderful experiences that I had here. And the first is, this is a wonderful paper. And this is the paper, and I'm going to actually, I really want to focus on the first two names, Noel Held, who is the first author on this paper, and also wrote a wonderful, and I tried to print it out, but there's a link for it, like a 2-page summary of the whole paper, and I'm a really big believer in that, being able to use that. But the second author is someone that I remember from the Carboniferous era when I attended the microbial diversity course in Woods Hole, John Waterbury. And he entranced me with cyanobacteria in general. And so what we're going to talk about today is a remarkable mix of so much material: basic marine ecology, Z diagrams and photosynthesis, glycogen provisioning and ballast of all things, elemental cycling, one of my favorite topics, and thank you, Elio, for this term, sociomicrobiology, and the strategies of microbes vast and various. I cannot emphasize what a cool paper this is. There's something here for everyone. And again, that summary essay by Dr. Held, I strongly recommend. And the first thing that I want to say, and I come at this as an educator. Oh, I'm
Mark,
sorry.
Please read us the title.
Huh? So sorry. I got myself excited. Thank you.
You did.
Okay, this is from Nature Microbiology, and it just came. Well, I'm trying to look for the date on this thing on it, but it just came out. I was very excited about it. And the title is Dynamic Diel Proteome and Daytime Nitrogenase Activity Supports Buoyancy in the Cyanobacterium Trichodesmium. And it's truly lovely work. I guess I'm flirting with the paper. I love it so much. So I'm trying to keep things on my screen and not seeing people. So again, just holler at me if I make an error or something. So what I often tell my students is, what are the two most important enzymes on Earth? And they always tell me DNA polymerase, right?
Rubisco,
which fixes inorganic carbon dioxide into organic compounds, and nitrogenase. And I love talking about nitrogenase with them because it isn't immediately apparent to them why biologically available nitrogen is so limiting. You say nitrogenous bases in DNA and RNA, and you say amino acids, and much becomes clear. This is a really good place to start at, because the most important thing that people don't get about marine microbiology is the ocean is essentially oligotrophic, very, very low in nutrients. And this is one of the reasons that you find photoautotrophs to the extent that you do. And the microbes that live there are remarkably able to assimilate elements very effectively. So nitrogenase is an amazing enzyme, and I think most people know this. It breaks that triple bond between the two nitrogen atoms, but it's the small masters that made this possible. It takes 16 ATP and 8 high-energy electrons to make that ammonia. And if we didn't have nitrogenase, the only source of biologically available nitrogen would be lightning strikes and volcanoes, which would limit the biosphere to some degree. The problem is nitrogenase is chemically damaged by oxygen. This isn't like Rubisco that gets confused. It's chemically damaged. And there are several strategies for avoiding this among the nitrogen-fixing organisms. And I did some of my PhD work on this. You can be an anaerobe, which is how it evolved, let's face it. You can have extremely rapid metabolism like Azotobacter. You can actually form a protective but non-functional version of nitrogenase, so it's protected from oxygen, but it's not working when that's the case. Or you can have symbioses, which of course I'm very interested in. So the oxygenic phototrophs that do nitrogen fixation have a problem. They're making the oxygen that damages the nitrogenase. So one approach is to separate them via time. And you see that in a lot of cyanobacteria where they'll make their oxygen during the day and they'll fix nitrogen at night. The other way to do that is separate them spatially, like our good friend Anna Bina does, with heterocysts and where you put the nitrogenase and exclude oxygen. So there are two players in this paper, and I just have to tell you that I remember seeing these things. And the first is trichodesmium, which has a wonderful nickname. It's called ocean sawdust or sea sawdust because of how it looks. It's a colonial organism of lots and lots of these different filaments together, these large floating colonies. And the other is Crocosphaera, which makes the more typical small spherical colonies. And one of the problems that all aquatic nitrogen fixers and photoautotrophs have is to stay in the right place in the water column. And some do it by swimming, yes, but most do it by having a kind of a fight between ballast, which is some biological polymer that they're creating, and their gas vesicles, which are microcompartments composed of proteins that contain gases, and they're not specific gases, sadly. And I remember my first microbiology professor, who I'm sure Elia remembers, Sid Rittenberg, demonstrated this in class when there was a mason jar that he had, and there was a layer of cyanobacteria, and he put a big cork in that so that it actually touched the water, and then he hit the top of the cork with a hammer. That increased the hydrostatic pressure inside the cells. The gas left the gas vesicles, and while you watched, that green layer sunk to the bottom. So the real trick here is to stay buoyant. And we're going to come back to that a couple of times in the context of this paper. And I am watching our time. So nitrogen fixation is extremely important. As 80 teragrams of nitrogen is fixed per year. And I didn't know what a teragram was, so I went and looked it up. It's a billion tons. And the really interesting thing about trichodesmium is it does something that's very counterintuitive. It fixes oxygen during the day. It fixes nitrogen, I should say, during the day, and also is producing oxygen via of photolysis and photosynthesis. Crocosphaera, on the other hand, does it more typically, as you might expect, making oxygen during the day, fixing nitrogen at night. So that's kind of what you need as you go into this. Questions or comments? Because I just don't want to yap at everybody.
Well, it's really about electron balance and how they're moving the electrons, and to give the bacteria their proper due. The way most people think about nitrogen is they go to their favorite big box store and they buy a bag of fertilizer. What they don't know is that bag of fertilizer took 1,400 pounds per square inch, 10 millibars, and between 400 and 500 degrees, Celsius, which is 752 to 932 degrees Fahrenheit, to fix the nitrogen. So bacteria are doing this at ambient temperatures, folks, at ambient pressures. I mean, they are the ultimate in chemical engineers.
Yeah. And it's just a lovely subject. I mean, I spent a lot of time learning about this in graduate school when I studied we called it rhizobium then. Now it's Cyanorhizobium mellilodi and alfalfa with the whole root nodule symbiosis. And it just always made me so appreciative of what goes on here. So what I want to draw your attention to in this paper is one of those heat maps that people like to do. What they did is they wanted to study how the proteome changes, and they're going to be looking at trichodesmium and Crocosphaera. And I have to say, I really hats off to the authors, because number one, I'm glad I can see colors. But number two, they actually do summaries of what the heat maps say in figure 1 below it a couple of times to help you. And on the left-hand side, you have trichodesmium, and on the right-hand side, you have Crocosphaera. And the important take-home lesson is, despite the sensitivity of nitrogenase for oxygen, in trichodesmium, they are clearly doing oxygenic photosynthesis and fixing nitrogen at the same time. On the other hand, Crocosphere is not. And they think this is so unusual, they did 3 separate proofs. Now, I think this is like, hammering that really seriously. So I don't know whether they got a lot of feedback that they didn't like initially. But the fact of the matter is, that's what they found. And I love the fact, because I'm getting older, that you can look at all of the proteins that are being made to figure out patterns and how things are regulated together in a way I never thought would be possible. So it's really quite a wonderful approach, and it fits what the 2 different strategies seem to have in mind. And it's interesting to me, because how do you control, again, where you are in the water column? And it turns out that a lot of microbes, when times are good, will polymerize glucose into what you and I call glycogen. It's just polyglucose, alpha 1,4 linked together, and the alpha linkage between them. And then they use it at night. Well, the problem is, if you're a floating organism, that glycogen has mass and will affect your ability to float. Getting more toward evolutionary form, also the form that you take is important to this, because you want to think about the effects of this on sinking. And that's the interesting part to this. So, once again, these organisms have a lot of gas vesicles. In fact, in Trichodesmian, 70% of the volume of that cell is made up of gas vacuoles. But they found out, as those colonies were sinking, the ones that sink the quickest had more glycogen. And that got the ghost of Darwin in my head thinking. Right? So what's interesting to me when you look at this is you can say to yourself, well, of course, you're a photoautotroph. You want to store up energy. You want to use it at different times. It takes you back to what Michael was just saying, and I know he'll want to opine, as the saying goes, on that in a little bit. But what I want to say about this is that it is very clear that Crocosphere makes it during the day and uses it at night to power nitrogenase. But Trichodesmium is using photosynthesis to power nitrogen fixation at the same time. And that's the part that's just remarkable to me. And it turns out that you seem to, if you look carefully at one of the most important genes in glycogen synthesis, you find out that that gene's expressed at night. They're not even making glycogen during the day. And that's where they actually do have quite a bit of commentary that fits in with what Michael was saying regarding energy balance, which is getting increasingly important to me.
And you can afford to sink at night when the sun is not shining, because you don't need to do photosynthesis.
The other thing is the microbes are stingy. They don't want to give away the electrons that evolution gave them the ability to capture photosynthetically. And they hold on to those electrons. And so it's the chicken and the egg question. What came first, the ability to fix carbon or the ability to fix nitrogen?
The answer is yes.
Yes.
I'm sorry. That's what I do in the classroom a lot, and they don't laugh. So that's okay.
Well, it's a hard question to answer, and it's really inherent in the energy flow and what's needed at the time.
When I talk about the Krebs cycle, for example, when I point out to students it can run backwards, and you can actually do reductive carboxylation that way. Students it really blows their mind, and they'll think, well, and I say, there's just not one cycle going on. You have lots of enzymes doing this. So some of them can go in one direction and some in the other. And that's why and this is just me, I think Krebs came first, but that's me. Because I have my questions about Rubisco. Anyway, getting back to it, I'm looking at the time. They spend some time talking, and this is relevant to what Michelle just mentioned, about Reynolds numbers. I mean, you have biologists, you have biochemists, you have marine biologists, and you have the physicists involved, because the shape that you take affects how quickly that you, quote, sink. And again, one of the things you find out when they did some of the proteomics work, they went and did it in the field. And this brought me back to my experiences at Woods Hole, because apparently somebody had to be picking the individual colonies by hand when they're doing field studies. You and I are used to just taking a volume of liquid and collecting it on a filter and doing whatever. You can't do that in the field. Well, you can, but that's not what they were doing. They were hand picking things 3 hours apart. So it's a Herculean effort. But at the same time, they really hammered at this. And I want to say parenthetically that there are some people who study trichodesmium who don't believe that there isn't some spatial differentiation where the processes take place, even though there aren't all the genes necessary for making heterocysts. You get a lot of argumentation, or nerd fights, as I call them. And that's great. That's how we advance. But it is 100% clear from this entire paper that trichodesmium genuinely is able to have coexisting in the same cytoplasm oxygenic photosynthesis and nitrogen fixation, which, is really hard to understand. And they do some discussion there, again, about balancing, frankly, where the electrons go. But it gets better. And it really does, because what Crocosphere does is it recycles its nitrogenase every day. It breaks it down and builds it up. And that's not what trichodesmium does. So there's actually a lack of iron. If you go to Wikipedia, you'll find out there's a whole entry on this organism, which is thought to be super important, as the major nitrogen fixer in the open ocean. And what's interesting is apparently trichodesmium can collect particles of dust as they're sinking and then utilize them slowly. And I draw your attention to figure 6, which is just a lovely bit, also in the Nature Microbiology essay that Dr. Hurd wrote. But it is 100% clear that trichodesmium is needing this iron, and so they're actually picking it up and slowly metabolizing it and making it available for others on their slow but steady descent into the darkness. And it is a remarkable paper, and I would love to hear what people have to say. I hurried a bit, and I apologize for that, but I do want to pay attention to time. As a college professor, that's what I do every lecture.
You don't want to go over. You get dinged for that.
I'll point out that they use incredibly sophisticated methods for this to get at what is really an elegant biological story that they lay out beautifully in their figure 6 schematic. So I encourage anybody who's looking at this paper, start with the figure 6 schematic, get that conceptual framework in your head, and then just marvel at how they were able to get in and measure, quantify particular components of this figure.
And not in just one area, but they brought together all of these different subdisciplines. And it was like seeing sparklers going off in my brain as I read paragraph after paragraph, because they would bring up other ideas that are worth pursuing just as they go. So I am so looking forward to hearing about the author, because I know that Michelle spoke to the author, because this is such a fantastic paper.
That's right. And Nicole Held was very excited to hear that it would be featured. She got her bachelor's degree at Stetson College in Florida, and then went and got her PhD with the senior author of this paper, Max Sato, at MIT and the Woods Hole Oceanographic Institute. And in the process, she went on multiple field studies to collect the samples. And I love just reading these fieldwork trips to places like the Mid-Atlantic Ridge to discover hydrothermal vents, and the Bermuda Ocean Time Series, and tropical Pacific, and et cetera, et cetera. So yeah, microbiologists aren't just sitting at the bench with tubes. This is really very cool biology. So she wanted to let people know that her path to science was not a straightforward one. She considered being a bioethicist, a physician, a diplomat, maybe a yoga teacher. But she really credits her mentor, Mac, who took her on when she was an unexperienced undergrad lab intern, showed faith in her and set her out independently on a research expedition only 2 weeks after joining his group. And that is where she really fell in love, found her passion for lab science and field research, and was committed. She also really appreciates the colleagues that she worked with. John Waterbury and Freddie Velos taught her everything she knows about keeping these fussy cyanobacteria alive and healthy. And evidently it's not so easy. She says they joke that it's a very dramatic microbe and requires a lot of attention. She also appreciates Matthew McIvan, who implemented all the two-dimensional mass spec methods that gave them the deep look into the proteomes, where they were able to compare proteomes at different times and from different species, et cetera. And that was tedious work. So one especially memorable moment during this project was after she'd been up all night sampling the cultures. One of her friends and office mates came with a very large cup of coffee for her. Mm-hmm. She still remembers that. Mm-hmm. But scientifically, she's remembering what an exciting day it was when they measured the glycogen content directly in the cells. And that really fit into the model that they were building. So it gave them a lot of confidence that they were thinking about this in an accurate way. So when asked if she has any advice for junior colleagues, she says her advice is to work with people you like. And I guess that makes sense if you can do it, because you spend a lot of time together. The other thing I was impressed, you, Mark, pointed out that she's a terrific writer and can make her science accessible. She has contributed a lot to science outreach. So she's given talks at a number of national meetings about how to apply to graduate school, but also at the MIT Museum Trivia Night. Ooh. And other public outreach events in the Boston area. So good for her. And I also wanted to point out that she has contributes to service. So she is on an advisory committee at MIT for planning their retreats, and also on the Presidential Advisory Cabinet and a committee on graduate programs. So this is a person who's got a lot of skills. And Noelle, we look forward to your career. She's now doing her postdoc in Zurich with Dr. Martin Ackerman. So let's see what's next for her.
It's such impressive work. I've just got to reiterate, and on so many levels. And it was so much fun to read. And I know that we could have dived in a lot deeper, but I'm glad to have had the opportunity to talk about a little bit of marine microbiology. Again, check out the Wikipedia page on Trichodesmium. It's really, you see all the pictures of it, and it makes me think. And there's some discussion even in the paper about the evolution of these. I'm not going to call them mats because they're not spherical. You get this idea that they kind of look, there's that beautiful picture. I don't know if Dr. Hurd drew it, but it's like the balloon with ballast and all the rest. They have so many different forms to them. They look so interesting to me. And I remember seeing them.
Mark, so the ferredoxin is the key here to getting energy to nitrogenase, right? And so they say they consume iron in the form of iron dust. So what is that? Where does that come from? Iron-rich dust. It's just floating around the world?
Yes.
Ferric hydroxide, rust.
And where is it? Where is it coming from? Any particular place?
The Earth's crust.
The crust.
Yeah. What's with the idea that it was floating around to seed the oceans with iron particles,
right,
to absorb CO2? What do you think of that?
Wow, that is since I love science fiction, I could see that going too far. And I think some people, didn't somebody actually try this with a couple of railroad cars worth of iron dust and did get a localized bloom?
Yes, that's right. I remember that. Michael, you talked about that a long time ago.
Yeah.
Yes.
But fortunately, it's the kind of, whenever people talk about engineering ecology, I get really nervous historically, right? But
don't go there.
I really love the idea because we tend to think, and I always thought of the ocean as being, rich and it's this wonderful nursery for life. But in fact, it's very low in nutrients and they're tricky. I have seen microbes that appear to be living on no added carbon source at all, and they're just living off impurities in the agar.
Oligotrophs actually will be here well after all the heterotrophs are gone.
Yeah.
The other cool aspect of this biology that we didn't have time to go into is it's not just a coordination of these different metabolic pathways, but actually the cell shape, and the shape dictates where the bacteria or the population can be in the water column, whether it's day and they need to be up there where the light is, and the iron-rich dust to capture. It benefits them to be in more communities, filamentous communities that give them buoyancy with these gas vesicles.
And it's thought to be providing the soluble form more effectively locally in that low nutrient environment too, not just for themselves, but for other organisms as well, which is just, altruistic of it.
So we have to go back in time to when the earth was first being seeded by the microbes, and consider the volcanic dust, because it was a very volcanic time, and a whole bunch of iron sulfur particles were being strewn out. Because remember, one of the great hazards of volcanoes today is the sulfurous gas and, the particles. And a lot of them are laden as iron sulfur particles that go out. And iron sulfur is just chock full of reducing power that the microbes will exploit. And so this really drives home the remarkable aspects that a microbe will not let any good energy source go until it gets its due.
One of the things that I have a trouble with students getting them to change their preconceived notions is they're very oxycentric. I've talked about the three centrisms before, but they think of everything as being a eukaryote. They think everything as being oxygen involved, or that all bacteria are E. coli, and none of those things are true, of course. But what's interesting is that when you get them to look at an anaerobic source of inoculum and they see things are zipping around really fast, even though they see that 36 to 38 ATP per mole of glucose in aerobic metabolism, but only like 2 net from anaerobic, that's what I tell them. I mean, microbes are good at the margins. They're really good at squeezing, because we're sloppy. We have too much energy. They're good at what they do. They've been Darwinoed to be really, really efficient about this.
Mm-hmm.
Yeah. The next faculty meeting, if you really want to impress your colleagues, bring in some rumen fluid and open it. You'll get many, many looks.
Yes, I will.
Where would I get such fluid, Michael?
Rumen fluid, you go to your favorite dairy that has a cow with a window. Yeah. And your good friend, Michelle, at Michigan probably
Patch Loss might be able to set me up.
He probably has a cow with a rumen window that you could reach in and dip out some rumen fluid. At one of my colleagues at work, Hal May, that was the first assignment of every new graduate student at Virginia Tech, is they had to go out to the farm, open the cow, and put the dipper in and dip out the rumen fluid to get the anaerobes.
I have to tell you, Michael, the way that they did it with me, and Michelle, this is for you, is you put on a glove that goes up to here.
Oh, no, you had to go the hard way.
Yes, I did. And then there's what is the name for it? It's an artificial fistula, but there's a fancy name for it. It's a one-way valve. And and I was holding a beaker, and I reach in, and then you're like reaching down, and you're scooping, and you're bringing the beaker out, and you're thinking to yourself, if you're a city boy like me, oh, this can't be good for the cow. The cow's eating while you're doing this. It's like,
it don't care. It don't care.
Boy, does it smell bad, but boy, is it interesting.
It's interesting, but it stinks that smell will go with you to the grave. Yeah. The minute you get a whiff, rumen fluid, where is it?
That's great. Well, this has been fascinating. Thank you so much, Mark, for telling us about this story. It's great.
I'm always happy to be here. Thank you so much for the invitation.
Really a great choice, Mark.
I really like having you on, Mike.
Thank you, Elio.
Thank you.
So that is TWiM number 259, show notes, microbe.tv/TWiM. And hey, listeners, I haven't gotten a question from you in 2 years. Are you distracted by the pandemic or something like that?
They're listening to TWiM.
Oh my gosh.
There's no time to write.
TWiM at microbe.tv. Send some questions in. We'd love to hear from you. And Microbe TV is now a 501c3. And so your contributions are tax deductible. So go over to microbe.tv/contribute and shed your excess money. Mark Martin's at the University of Puget Sound. Thank you, Mark.
Thank you.
Michelle Swanson's at the University of Michigan. Thanks, Michelle.
My pleasure. Good to see you all.
Now, football is almost over. Is that right?
Football is over. Yes.
It's over. Okay.
But our women's basketball team is up to 5 in the country.
Okay. So I guess football didn't go well.
It did. We won the Big Ten championship and went to the national semifinals.
Oh, gosh. I had no idea any of this was happening.
Yep.
I'm distracted by COVID.
You are.
Elio Schechter is at Small Things Considered. Thank you, Elio.
My pleasure.
And Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was, no, it's not TWiM. This episode of TWiM was edited by Ray Ortega. How does the saying go? Everything looks like a what to a what? You know what I'm talking about, that saying?
Yeah, I do.
Everything looks like a nail to a hammer.
That's
right.
Hammer. There you go. I don't think that's quite it, but you get the picture. To me, everything looks like a TWiM.
Yes.
Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.